pleased to introduce Dr. Christine Ambrosone, our Saxon Graham Lecturer for 2025. Christine is the Jane and Phil Hubble Family Endowed Chair in Cancer Prevention. She's a distinguished professor of oncology and Senior Vice President for Population Sciences at the Roswell Park Comprehensive Cancer Center. She's also the director of the Roswell Park Biorepository and Laboratory Services Shared Resource. This woman, her scholarship is outstanding. She has over 400 peer-reviewed manuscripts, including works in high-impact journals like the New England Journal of Medicine, Nature Genetics, and JAMA. Her work focuses primarily on breast cancer disparities, where she investigates genetic and non-genetic factors related to aggressive breast cancer in Black women. She also studies how lifestyle factors, as well as genetics and tumor characteristics, are associated with treatment outcomes. Christine has been continuously funded by the National Cancer Institute and has served on the NCI Board of Scientific Advisors, as well as holding other advisory and leadership roles in the field. And I want to share with you, it's also a great personal pleasure for me to welcome Christine to this lectureship. I've known Christine since we were graduate students together. I value her friendship and am really sincerely inspired by her contributions to science. She's always been a really strong and independent thinker, and I look forward to her talk this afternoon. So I'll say I'm very proud to introduce Dr. Ambrosone, an exceptional scholar and an amazing leader among our distinguished alumna. She'll speak to us today on the use of molecular epidemiology to understand cancer risk and outcomes. Please give a very warm welcome to our 2025 Saxon Graham Lecturer, Dr. Christine Ambrosone.
Wow. Thank you so much, Pauline, for that very warm and generous introduction. I'm very happy to be invited and grateful to be asked to do this lectureship. I graduated from UB. I got my PhD here with Roswell Park. I've been mentored by a lot of the faculty here over the years. So I'm just thrilled to be giving this talk in honor of Dr. Saxon Graham, one of the, as you know, one of the first people really to do molecular nutritional epidemiology, and he was a real inspiration to me. I was there for maybe a couple of years when Dr. Graham was there, and we all really looked up to him. But he was kind and he was warm, as well as being such a great scholar. So he studied diet and cancer, and when I came in, there was a lot of data. He had gotten NIH funding for a series of case control studies in different cancers, and as well as a large cohort study. So we were like in this wonderful palace of resources and data with all of that. We didn't really have to even think about it. It's very, very rich. He also mentored others who carried on that legacy of nutritional epidemiology, including Drs. Jim Marshall and Dr. Joe Freudenheim, both of whom were my mentors. I started it when EEH was the Department of Social and Preventive Medicine on Main Street, and interacted with Saxon and with Jim and Joe. So as I learned epidemiology, and it became clear how really complex it is and how little we really know. Because as you think about it, for some cancers, there are very strong relationships between risk factors and the disease, like smoking and lung cancer, alcohol and esophageal cancer, HPV and cervical cancer, sun exposure and skin cancer. But for many cancers, the risk factors we know don't really explain a large portion of the variability. And I've been studying breast cancer, and this is even more a problem for breast because of the role of hormones in breast cancer. So it's hard to tease out other factors. So why is it so hard to explain more of cancer or cancer etiology and epidemiologic studies? Well, probably a big part is study design issues. Can we really measure the exposures so that represents it well? Misclassification of exposures, misclassification of disease, actually, because there's different molecular characteristics so that tumors are not all the same. And recall biases, especially for diet, when you're asking people, what did you eat 2 years ago? And other studies, even trying to get back to adolescence or young adulthood. So recall bias is a problem. And then sample size is never big enough for the variables that you're looking at. And then timing of exposures. Some exposures may have occurred during adolescence or early adulthood. It's very hard to pin these things down. So to ask people about things they're exposed to in the past is really, really problematic. And then the area that I'm really interested is in heterogeneity within study populations, in a lot of exposures, in susceptibility, and alterations with tumors. Good example is smoking. We know that smoking causes lung cancer, but only 1 in 10 smokers get the disease. So why is this? And for a number of years, people have been interested in potential differences in susceptibility. And for a number of years, we could only look at family history as probably genetic susceptibility. But going on, trying to understand this, it's really been the basis for molecular epidemiology and the field that I'm really excited about still after 20, 25 years. I came across this in a text from 1956, Biochemical Individuality, where he comments everybody has their own metabolic characteristics, and applications of biochemistry, and I would say epi, to human beings must take these differences into account. And goes on, all diseases are related to biochemical individuality. And 1956, he was talking about this, and we didn't really take this into account in our research studies until, I don't know, maybe 15, 20 years ago, we started thinking about it. And much of this variability comes from genetic polymorphisms. If people aren't familiar with this, all of our enzymes, protein activities are encoded by genes, and there are common variants in like 20% of the population, 30% of the population that cause our bodies to react differently or process things differently. So this has been an area of investigation for a number of years. It kind of rose and then kind of fell, and now looking at genetic variants and big genome-wide association studies. But I am interested more in these functional polymorphisms. And in 1996, I took a job at the National Center for Toxicological Research with Fred Cadlebar, and he was a biochemist, and he was the director of the Division of Epidemiology, and his idea was, well, let's go get those livers out of the freezer and measure, metabolism or whatever. And I said, no, Fred. So we really had to bring together these areas, the basic science, biochemistry, toxicology, with molecular epidemiology. And I was telling students today, he was a fabulous mentor. We were at a meeting. My first abstract got rejected from Society for Epidemiologic Research, and I was kind of whining about it at a meeting, and he said, we should have our own group. We should have a molecular epidemiology group. So Fred just charged on. We started it. It became eventually part of the AACR, but he had done work in aromatic amines, especially in meat that's cooked at high temperatures. So I went there really to learn this area because I really knew nothing of biochemistry. One, I've got that picture of the woman with the vegetables to remind me some of the best advice he gave me, and that I think I can give other people, is to think of your research as a farmer. And he said, you've got multiple fields. One field, you're just planting the seeds. Other fields, you're nurturing them, you're watering, you're weeding, and then you have fields that you're going to harvest. So if you think about your research, you have papers you're writing now. You have other work ongoing that will lead to papers in a couple of years. And then you have studies or projects that you're just starting. So you need to be thinking about 5, 10, 15 years from now, what kind of research am I going to be able to write up? So very, very great advice. So thinking about susceptibility and how that might interact with exposures, I was studying with Joe Freudenheim and with Jim Marshall, and really interested in diet and the role of diet in cancer etiology, and especially for breast cancer. There's very inconsistent findings in case control studies, no association in the Harvard Nurses study or the cohort pooling project. So I felt in my core that diet and vegetables and fruit have got to make you less susceptible to cancer. It's got to reduce risk of cancer, but we're not seeing it in these studies. So why is that? And I wondered if different responses in different groups could obscure these clear relationships. And we used the Western New York Diet Study that Saxon had started and looked at family history in relation to dietary antioxidants. And we did see differential relations according to whether or not the person had a family history of breast cancer or not. And then when I went to National Center for Toxicological Research to really learn from Fred Kadlebar, I learned a lot about carcinogen metabolism and cancer risk. And this can apply to many carcinogens, not all, but many. Cannot readily be detoxified, but they're first activated by the cytochrome P450 enzymes, phase 1 enzymes. So they're reactive and they're looking for something to bind to. And these are generally detoxified by the phase 2 enzymes, glutathione transferase and acetyltransferases, et cetera. But if they're not, these reactive intermediates can bind to DNA and cause DNA damage. Our body has great systems to prevent. I mean, if we didn't have these checks and balances, we'd all be dead from cancer years and years and years ago. But the body really can take care of this stuff. And there's DNA repair that can get rid of those DNA adducts, get rid of any damaged cells. But if the cell does get initiated, can ultimately lead to cancer. And those polymorphisms are in all of these genes, all of these pathways. So we all have variability in how we respond to the things that we're exposed to. And actually, the way I met Fred Kadlebar was I was on my dissertation project. We were looking at whether or not smoking was related to breast cancer and if differences in metabolism could make some people more susceptible than others. And Fred Kadlebar was an expert in that area, so I had contacted him to get his input. And we were talking about this morning with the students, like, don't be afraid to contact people. If you want information, if you want some help, we ended up him hiring me, and we did a lot of work together. So reach out to people, and don't feel bad if you get a little rebuffed, because most times it will be successful. So we then started looking at mechanisms. I went to a meeting in Buffalo on antioxidants. So it was actually called free radicals. And we all joked about, the 60s, the radicals. But it was related to oxidative stress. And it really opened my eyes because I hadn't thought about this. And a lot of cancer risk factors can be linked through oxidative stress. And we can use genetic polymorphisms in those pathways as surrogates for systemic endogenous capabilities. So I was contacted by this student at Cornell who wanted to do her research with me, her dissertation. And Ji Young An came for a few years, and she was just an incredible postdoc, student and then postdoc. And first we used data from the Western New York Diet Study, and then collaborating. We were talking today about making collaborations with people. Long Island breast cancer was a very large study of breast cancer with very rich data and DNA for genetic studies. And so we collaborated with Marilee Gammon and Regina Santella on that study. And I'm not going to get into this, but there are enzymes that generate reactive oxygen species, superoxide dismutase, myeloperoxidase, but the glutathione transferases can block those, and also catalase and glutathione peroxidase. So these pathways that can generate or quench free radicals. These enzymes are polymorphic, and SNPs could serve as surrogates for these endogenous capabilities. So I'm not going to go through all of these. I wanted to talk about this because of the Saxon's work and the impact of diet. So catalase reduces hydrogen peroxide, and there's a variant in the promoter, greater transcription with C allele, so there's less ROS. And we were interested in whether or not there are relationships between genotypes and activity, and also with risk, and is risk modified by dietary antioxidants. So up on the left, we had samples so that we could look at activity of catalase in blood samples. We had a few hundred people, and then genotyped. And those with CC genotypes had the highest activity, CT lower, and TT the lowest activity. So high activity is going to reduce oxidative stress, low, less so. And Ji Young published that paper. And then in the Long Island breast cancer study, we looked at genotypes and risk. And here we see a slight reduction in risk with those CC genotypes that reduce oxidative stress. But then we looked by diet, and this is just two components. We pulled out carotenoids and ascorbic acid. The yellow is people with low intake of fruits and vegetables. You see a reduction in risk, not that much though, with the CC genotypes, but for people who had diet rich in antioxidants, there was this great interaction with those genotypes, so very, very strong reduced risk. I really loved this paper because of the phenotyping and the risk. So using molecular epidemiology, we can see that dietary factors influence breast cancer risk, even among those who are susceptible based on their genetic makeup. Then when I was in Arkansas, now I'm going to turn to the work that I've really been doing for the last 20 years. I met Debbie Irwin, and she was the founder of the Witness Project. And here women who were survivors of breast cancer and cervical cancer, Black women who lived primarily in the Mississippi Delta, they would go into churches and they would witness about self-examination, early detection, and preventing mortality from breast cancer. Because when I was in graduate school, I didn't really think about breast cancer in Black women because it was less common in Black women than in white women, but they do have higher mortality. So I just became very aware of this when I was in Arkansas and working with Debbie Irwin. And this just shows rates of incidence, and you can see back around 2000, there were higher rates in white women, but those are coming down, and lower rates in Black women, but those are going up. And on the other side, we've got mortality. We see nice reduction in mortality for both groups, but we still see higher mortality for non-Hispanic Black women. Oops, am I right? Yeah. Okay, so why is this? Why do we see this higher mortality in Black women? Well, the first thing we'll think about is socioeconomic and societal factors. Especially when I was in Arkansas, there were women living in the Mississippi Delta who did not have cars. There's no public transportation. You're not going to be going for mammograms every year. There's later stage of diagnosis, poorer access to optimal care. We see this a lot in the literature for a number of reasons. Less adherence to treatment, some because of comorbidities, others because of life getting in the way. But then the one thing that we were really interested in was tumor biology, because Black women are more likely to be diagnosed with tumors that are high grade, more mitotic activity, and negative for estrogen receptors. So estrogen receptor positive breast tumors can be treated with anti-estrogens, and you could see much better cure rates, whereas ER negative, until more recently, there really haven't been any therapeutic targets. And these are also more aggressive tumors. And the rates of triple negative breast cancer by race, the top one is non-Hispanic Black women. And you can see those rates are so much higher for Black women than they are for white women or for the other groups. So why is this? What's driving these differences in tumor subtypes? Is it biology? Is it genetics or epigenetics? Or is it epidemiologic risk factors? Because if it's risk factors, we can intervene. We can probably do something to prevent more aggressive breast cancers in everybody. Trying to think this through and reading, we looked at risk factors in different groups, and Black women on average have earlier age at menarche, more children at an earlier age, and are less likely to breastfeed than white women. So we were wondering if a high hormonal milieu, higher circulating estrogen levels could kind of downshift those risk factors to a younger age and be associated with early onset ER negative breast cancer. To look at this, we initiated the Women's Circle of Health, first funded through a DoD center when I was at Mount Sinai, and then additional grants. We had a program project grant. Elisa Bandera and Chi-Chen Hong had an R01 to look at treatment outcomes. And then the Breast Cancer Research Foundation has funded me for a number of years, and also a gift from the Hubble family. Oh, okay. Whew. So we worked with, first we enrolled through hospitals in New York, and then we worked with the New Jersey Cancer Registry, which is the SEER site, and enrolled cases, detailed in-person interviews at home. We did body measurements, initially collected blood, later saliva for DNA extraction. And what's really feeding our research is that we got consents to get pathology reports and tumor blocks of remnant tissue from the hospitals so that we could really look at what's going on in these tumors. Well, as we were collecting data, which takes years and years and years, there was this paper published by Sarah Sukumar's group looking at DNA methylation in relation to breast cancer in younger Black women. And they just looked at a few genes, and they saw that in tumors that were negative for estrogen and progesterone receptors and women who were less than 50, they had significantly higher frequency of methylation in Black women in the majority of genes compared to tumors from white women. This really got the wheels turning because we were collecting data. We were getting tumors. We could investigate this at a larger scale than just looking at a few genes in 100 or so women. So we got a grant. I worked with Mike Higgins at Roswell, who is a basic scientist. He works in epigenetics and really knows what's going on with methylation and changes to DNA. So we used a genome-wide approach, 450K array, and we had DNA from more than 700 women, Black women and white women. And our results were similar to Sarah Sukumar's group. The majority of differentially methylated loci by race were in ER-negative breast cancer. And when we went to look at the most differentially methylated by estrogen receptor status, this gene FOXA1 came up. And especially in tumors from Black women, ER-negative versus ER-positive. And we also saw on the bottom, you can see reductions in gene expression associated with the methylation. And FOXA1, I didn't know anything about this gene, but it turns on response elements in estrogen receptor genes, and it's very highly involved in making luminal cells from luminal progenitor cells and preventing basal-like or triple-negative breast cancer. So again, this is how science works. You're doing your focused research, and then you see publications. You think, wow, is this relevant for what I'm doing? Can we look at this? And there was a paper published by the Carolina Breast Cancer Study in 2008 looking at hormone receptor positive tumors and triple negative breast cancer, negative for ER, PR, HER2. And they looked at having children and breastfeeding. And with with the reference group, women who did not have children, they looked at those who had 1 to 2 children or 3+, among those who breastfed and those who did not. And you can see for hormone receptor positive, you see a reduction in risk with having children. Doesn't matter whether these women breastfed or not. But for triple negative breast cancer, almost a 2-fold increase in risk for women who had children and did not breastfeed. If they breastfed, the risk went much, much lower. And this was published in Breast Cancer Treatment Research. It's an okay journal, but it's not a well-recognized journal. And I was friends with the author, Bob Miller, and Bob, why is this buried in that journal? This should be like New York Times, from a high-impact journal. Everybody should be aware of this. If we could reduce risk of this deadly triple-negative breast cancer by encouraging women to breastfeed, that's really huge. So we got together with also the Black Women's Health Study and also in our study and looked at this, and we saw the same relationships. But nobody was aware of it. I gave talks at AACR, big scientific meetings, and asked people who's aware of this, that having children increases risk of ER-negative, triple-negative breast cancer, and breastfeeding reduces it. Very few people. And again, can't get the papers into high-impact journals. So Bob Milliken, Julie Palmer, who is the PI of the Black Women's Health Study, and I were at a meeting, and CI wanted us to pool our data and do a genome-wide association study. And we're like, these are like little polymorphisms that increase just a little bit. We don't really care about this. Let's try to get with a really big sample size. Let's nail down this relationship between parity, breastfeeding, and ER-negative and triple-negative breast cancer. So we went back, we did a real hurry, and we developed a program project grant. We formed the AMBER Consortium, pooling data from 4 studies of breast cancer in Black women, and we collected tumor blocks, did subtypes. And we had 4 projects, and the first one was relationship between parity breastfeeding and those breast cancer subtypes. And our findings were confirmed here. You can see those 2 top odds ratios. These are women who never breastfed, ER-negative in the blue, ER-positive in the red. We see increased risk with number of children, but we see a reduction in risk of ER-positive, whether women breastfed or not. But it's in that ER-negative where you see breastfeeding really having the impact. So thinking through this, reading the literature, especially where ER-positive or ER-negative tumors come from. There's a literature on luminal progenitor cells going to either luminal tumors or basal-like tumors with multiple events. So we wondered, well, could having children and not breastfeeding be key events that lead to this FOXA1 methylation, and could that be involved in driving these ER-negative and basal-like tumors? So we looked at this in the Women's Circle of Health study, and small numbers, but you can see we looked at parity versus nulliparity, number of births stratified by breastfeeding, and the really big hit for FOXA1 methylation is for women who did not breastfeed. So with that data, we got together those 3 studies, and we obtained funding for a very large array for a large study of methylation subtypes and risk factors in Black women who are more likely to have children and not to breastfeed. So we had a large number of ER-negative and ER-positive tumors with methylation data. And I'm not going to get deep into this, and I should have said in the beginning, don't worry about getting the details. I'm just trying to get across these bigger concepts of mechanisms and how they work. So here we looked at DNA methylation in relation to having children and breastfeeding. And those lines on the top are the statistically significant lines. There are a few hits on some of the chromosomes, but really nothing that's significant among women who breastfed. But for those who did not breastfeed, you see these hits that are almost significant, and then above the significance level, and this is with multiple comparisons. And a number of them are in FOXA1, and this is what we'd seen in our smaller study. We are very excited about this because as epidemiologists, you love to see replication of your results and your data. Think a good journal was interested. Oh no, you've published on this before, even though it was a smaller sample set. And it was like, well, who knows if those results are true, but here we see replication. Now I'm going to switch over to kind of a second part of the talk. And Kunle Odunsi and Calvin Lee were immunologists who worked at Roswell Park. And we had fun together. We liked each other. And so we started getting together after work every couple of months in the hotel bar attached to Roswell Park. And we just kind of chatted about our research and what we were working on and thinking about. And I was saying, well, I'm interested in aggressive breast cancer in Black women, and prostate cancer. We see similar things. Black men are more likely to have high-grade, more aggressive prostate tumors. And Kelvin and Kunle were like, oh, that's a no-brainer, of course. We got different immune systems in people with African ancestry than people with European ancestry. So we worked together and we developed, this was a project in that PO1, that adaptation to endemic infectious disease in sub-Saharan Africa could result in a more robust immune inflammatory response. And this increases likelihood of surviving through reproductive years, but could be maladaptive in a Western modern society. So we wondered about relationship to cancer. Could this be related to more aggressive diseases? And a group at Columbia many years ago, Don Hirschman and Al Nugget, were looking at the Duffy antigen receptor for chemokines, which is only in those of African ancestry, this polymorphism. And they were looking at it in relation to response to therapy. This is really interesting that people who have that Duffy null don't seem to respond as well to treatment, and they get their blood counts go really low. And it wasn't until a lot of attention to this that clinicians realized, oh, their normal set point is lower than white populations, European populations. So they're not neutropenic when their counts go lower. So I was very aware of that. And what accounts for this is this Duffy antigen receptor for chemokines, the dark gene. There's a literature on these differences that now that after our project, immune responses differ by ancestry. Populations in differences in immune response are under genetic control. and genetic variants associated with these differences are enriched for natural selection signatures of human adaptation. So with our project here, we measured chemokines and cytokines in blood from controls from our studies. And we looked at a number of them, and we saw differences. Some were higher in European Americans, some were higher in African Americans. And when we adjusted for epidemiologic variables, most of those associations went away, except for CCL2 and 11, IL-10, and INF alpha 2. And Chi-Chen Hong and Song Yao led this work. So this suggested a strong genetic component. And here, Song went on and looked at differences in circulating cytokines by quartiles of European ancestry. And the gray bar on the left, it's the lowest up to the highest levels of European ancestry. And we see this very strong for CCL2, CCL11, not for the other two. And Song did admixture mapping to see if you could really get a signal. Where is that coming from? Where are those differences coming from? And for both CCL2 and CCL11, identified one peak for both of them. And lo and behold, it's the Duffy antigen receptor gene. The dark gene is controlling these chemokine levels. So here, looking at levels by the dark genotypes, the European Americans are over on the left in the gray. And then by genotype, those with CC genotypes for the dark gene have the lowest levels of CCL2 and CCL11. So population differences in cytokines, many accounted for by lifestyle factors, strong ancestral component to the serum levels, and that variant in the Duffy antigen receptors responsible for ancestral differences. It's null in blacks and results in a loss of protein on red blood cells. So Nisha Nair was a graduate student here, and she worked with us for a while. And I really wanted to look at this polymorphism in relation to breast cancer risk, because there's a group who's been claiming with very small sample size and very heterogeneous populations that the dark gene plays a big role in ER-negative and triple-negative breast cancer in Black women. And I assumed that we would replicate this. So we looked at genotypes, and we looked at levels of T cell density in relation to these genotypes. And you can see for TT genotypes that they're higher, and this is true for overall breast cancer, for ER positive, and for ER negative breast cancer. And then we wanted to look at it in relation to risk, and we didn't really have the sample size, so we collaborated with Wei Zhang, who has a consortium that we participate in, large number of Black women enrolled with breast cancer. And here we looked at the genotypes overall, and we see a reduction in risk, not statistically significant, just borderline for those TT genotypes. For ER negative, we don't really see that at all, maybe because sample size is smaller. And triple negative, we don't see that. On the other hand, we see a suggestion of increased risk. So we've submitted this paper because it very clearly, with large sample size, shows that Duffy antigen receptor plays a role in a lot of things, but likely not more aggressive breast cancer. So this took us back to our key question: could prolonged and heightened inflammatory response in Blacks lead to tumors co-evolving in this milieu to evade immune surveillance, resulting in more aggressive tumors? And Angela O'Million in our group had a big intellectual role in this, in thinking through this issue. So we looked at tumor infiltrating lymphocytes in tumors from women in the WCHS. So before we had looked at cytokines and chemokines in blood levels and in serum, here we're looking at the tumors. And do we see more immune cells in tumors according to genotypes? So Thayer Corey is a pathologist at Roswell. This man is so dedicated. We have had thousands of cases in the Women's Circle Health Study and now the North-South Breast Cancer Study. He takes the sections, he looks at them, he grades them, he gives us information on what he's seeing, and then he circles where we can take cores that we can use for research. And he was doing this. He said, you know, some of these look very different from others. There's a lot of infiltrating lymphocytes. And when we stratified, when we got the data all together, we could see that these higher levels of TILs were primarily in Black women, and didn't matter if they were hormone receptor positive, we saw the difference, ER negative or triple negative. We still see higher levels of TILs in Black women than in white women. And this was kind of confusing because we know that Black women have poorer survival than white women, and TILs, higher levels of TILs are associated with better outcomes. So we looked at survival according to TILs, and yes, we did see that higher levels were associated with better survival, both overall and disease specific. But in this population, where we looked at differences between Black and white women, with the solid line being Black women, again, we see poorer survival for Black women than for white women. So what's going on? We know TILs give you better survival. Black women have higher TILs, but they've got poorer survival. So Song Yao did a lot of work getting, identifying the types of immune cells and gene signatures, and found a combination that were predominantly exhausted T cells. And exhausted T cells were associated with poor survival. And here we can see, we look at ratios of total CD8 T cells to TILs, CD8 cells to exhausted, and to Tregs. And for Black women, we see lower proportions, so more exhausted T cells. And I'm not going to go through all the details of this, but they're associated with poor outcomes, and this was in our study, as well as in the TCGA, which is a national collection of tumors. And then on the bottom, we look at survival adjusted for age and clinical prognostic factors. And the red bar is CD8 low, and the exhausted T cells high. And those are more predominant in Black women in WCHS and in TCGA. So we've been going on here trying to really better understand the immune contexture. And Tina Darabnush-Tehrani, who's a graduate student here, now she's at Roswell. She was a student here, and now she's with us. And she's been working with Angela O'Million, and Ricky Caniato is doing this work too, trying to look at multiple immune markers in tumors. And we're just in the early stages of this. And Tina's done tons of work going through trying to identify specific types of T cells and quantify them. So our goal then is to look at T cells and exhausted T cells, other types of T cells in relation to survival outcomes. We've got a new study that I'm really excited about. Karen Hume here in the audience has been coordinating this whole thing. And we've been working with the New York State Cancer Registry and the Louisiana State Registry, calling it the North-South Breast Cancer Study for lack of a better name. And we're getting women who are diagnosed with breast cancer and contacting them over the phone. We're consenting them on the phone and then doing interviews and getting releases for the tumor so we can get them from the hospitals. And we've enrolled 2,700 cases as of April 2nd. We've probably got more now. And the tumor blocks are rolling in. We've got more than 1,500 tumor blocks from more than 1,500 people. So this is our next step, is to really look at what's going on in the tumor, especially looking at immune contexture. And is this associated with more aggressive disease and outcomes? Ricky Caniano is very interested in modifiable risk factors. Does physical activity increase levels of good T cells? are there modifiable factors that can really improve the immune contexture in tumor cells? So this is an exciting new area that we're going into. So we're going to expand on these subsets and the tumors from Black and white women using some newer technology. In addition to looking at survival outcomes and physical activity, we're collaborating with experts on neighborhood and stress, and we're going to be looking at those factors in relation to immune contexture. Annette Stanton, who's at UCLA, and Neha Gall, who's at Memorial Sloan Kettering. So very exciting times ahead. Every now and then I'll think, oh, maybe I should retire. And then I think, oh, there's so much exciting work still to do. So this isn't really an up-to-date slide. There are so many people who have been involved in all of this work in the AMBER Consortium: Elisa Bandera, Chi Chen, Song Yao, Julie Palmer, Andy Olshan, Melissa Troster, and then colleagues from the University of Hawaii. The tumor analysis, we've talked about and acknowledged many of these people at Roswell. David Chang was a postdoc here, and then he moved on to Ohio State University. And the Breast Cancer Research Foundation, they invited me to submit a proposal in 2008. And I just write a little renewal every year and get funded. This is like manna from heaven. This is such a gift because you can get a lot of pilot work done. Like NIH grants are very hard to get, and the reviewers are picky, rigorous. Let's say the reviews are very rigorous. Breast Cancer Research Foundation, yes, they approve it if it's solid, and it gives us the funds to be able to go on and get good pilot data to then get more national funding. So I guess I will stop there. It's a really exciting time for cancer research, and we're all worried about what's going to happen with funding and a lot of other things, but we're just going to keep plugging on, just keep going on. So thank you very much.
Thank you, Christine. That was a marvelous lecture. And now we open for questions. So Jen is going to be on the other side of the room with the microphone. I'm over here with the microphone. If you want to just raise your hand, we'll come to you so you can ask questions. Here we go.
That was terrific, Christine. Thank you so much. It's really exciting to see it all together, all the different parts you're doing. I'm wondering, in the more recent work, are you including breastfeeding as, you found those stark differences with the tumor, the TILs and the other things. Do you see differences by breastfeeding?
Well, we haven't looked at that. Really, we're really just starting to get the contexture in the tumors, and we haven't started looking at the risk factors. This is in grant proposals, but yeah. Usually during the night, I'll think, oh, we should look at this or look at that. I'm sure you all get those little epiphanies, and then you forget when you wake up. But
Hi, Lorraine Collins. Thank you for a very interesting talk. I also wanted to ask about breastfeeding because one of the things we know is that African American women have lower rates of breastfeeding. Is there any possibility of sharing kind of the preventative aspects of breastfeeding with regard to cancer as a way of boosting those rates?
Yes.
Because that's important information, and I have to say I haven't heard it before.
Yeah, absolutely. And we've really tried to get the word out. We put in a grant proposal to specifically look at breastfeeding, like how long do you have to breastfeed to get that effect? How many children? The last one, the first one, and NCI didn't even accept it for review because they said it's not relevant for cancer. I was fuming mad. I usually don't react to, grant losses, but I was not happy about that. But we worked with Debbie Irwin from the Witness Project, and they have put it in their curriculum now. So when they're working with populations to increase breast self-exam and screening, they're also putting in the information about breastfeeding. But I'm not really sure how else to get it out. And this is, when I started thinking about that, wow, usually you publish your paper and you put it out there, but we really need to try to get the word out if you can reduce triple negative breast cancer in Black women. Yeah, thank you.
So I have a question. Um, I'm really interested in the immunological pathway, and I wonder if you've done any, or if you're able, probably not, because you can't really do flow cytometry or anything, but can you separate T cells? Can you see what you've got, like Th1, Th2 ratios, looking at Th17? Can you get a picture of the inflammatory nature of the T cells or not that you've got kind of going on there?
Yeah, this is actually something that Tina is working on. And do you want to say something?
But Jen, give you.
Sorry, I didn't get the first half of your question, but like being able to tell the different immune cells. Usually when we like the panels that we have different staining for different immune markers. So we have a multispectral image where we can like turn off and on the immune markers that we want to see. So like if we want to see the T cells alone, we can turn that on and like everything else off. And yeah, so we have like clear images of our immune markers in the cores.
So you can see the T cell subtypes. I guess that's what I'm asking.
So if we stain for like different T cells, so that's what I'm doing with the B cells.
Gotcha.
We have different B cells, like the naive B cells, like IgDs, like different B cells and T cells. We could see them, yes.
Great. Thank you.
No, it's okay. I
have
had some extra slides, but I can't find them that show some of that work with the T cells.
Me?
Hi, Dr. Ambrosone. I'm a member of the Saxon Graham family. I'm also a retired pediatrician and former faculty member at Children's Hospital of Buffalo. I have an idea about how you could get the word out about breastfeeding and reduce risk of triple negative breast cancer in African American women. Perhaps you could work with the State Department of Health. We'll reach out to all the hospitals in the state. Prepare some kind of statement, brochure, whatever you want to call it, that these hospitals can use in their maternity wards and hand that out to the African American mothers.
Yeah.
I think that would be very helpful. If you'd want to take it a step back, you could even work through the state-funded OB clinics and get the word out there. But it's got to be out in the community, into the places where these women are getting prenatal care and then giving birth, to make it more effective.
So Debbie Irwin, who led the Witness Project, she worked with us on that, and she obtained a grant. But it never really got down to testing whether you could change behavior or influence decisions. It was looking more at how best to present the data. So she did work some with the hospitals, but yeah, I regret not going farther with that, actually, because I feel like we could make an impact.
So.
Hi, thank you for your talk. I'm Marina Filer. I'm a early faculty in EEH, so nice to meet you. So everything you do is already very complicated, so not to add kind of another layer of complexity, but I'm an environmental epidemiologist, so I'm always thinking from that perspective. So the work I do really looks at environmental exposures and immune dysfunction in children. And now I'm kind of starting, not just me, but everybody in the field is really starting to think of environmental exposures also just as health inequities. We do see that, Black communities with higher poverty, they just have higher environmental exposures in
Mhm.
addition to all the other, social determinants of health that they experience. So it's
Mhm.
just almost considered another health inequity.
Mhm.
There's also kind of new compelling evidence that environmental exposures reduce the ability to breastfeed, just breastfeeding duration. And so I know maybe, you're not aware of this, it's a different field than yours, but if you've ever kind of considered how maybe environmental exposures are impacting any of these pathways, especially considering all the, major differences between Black and white women.
Yeah, yeah. So the Breast Cancer Research Foundation had a health equity initiative that started 2 years ago. And they're working with our study, the Women's Circle Health Study, the North-South Breast Cancer Study, the Black Women's Health Study, and the Carolina Breast Cancer Study. And they brought everybody together. We kind of let go of all of our, competitiveness. And we're putting some of the same measures into our questionnaires and to look at exactly like you're talking about. And there are folks who are very interested in the environment who are just coming in, and we'll see where that goes, because I'm like, whoa, it's too much. But that's why you need a lot of collaborators with expertise from all over. Thanks.
Thanks for your talk. I actually worked with Debbie Irwin on her R21. Renee Cadzo used to be at DuVille, now at UB.
Yeah.
And just for those who are asking questions about the messaging related to cancer prevention versus kind of your traditional messaging for breast cancer or breastfeeding, we kind of compared if you give them the standard messaging versus you can reduce your breast cancer risk. We didn't see a difference in breastfeeding uptake in that study. We just saw that social support really helped with breastfeeding uptake. So, I mean, I guess not terribly surprising. But on a plus side, there's a number of organizations affiliated with the coalition locally that are trying to increase breastfeeding among Black women through the training of doulas and breastfeeding peer support workers. So there is lots of good work going on with that. My question was, and I may have read this before, but I can't recall. Do you see the same disparities by race when you look at countries that don't have the same level of systemic racism that we do in the U.S.? So I don't know if that research has been done or the data is available, but I suspect that stress is, as your colleagues are looking at, is a big player here. So it'd be interesting to know if those disparities exist in other countries.
Yeah, there is part of this health equity initiative that BCRF has. Melissa Davis has collaborators. She just got a huge grant. I can't remember who it's through, but she's collaborating with a few partners in Africa, and they're really trying to look at breast cancer in African women, and do we see the same differences as we see here? And I think that rates are lower, although you don't have as good registry capture in Africa. But it looks like rates of ER negative and triple negative are lower than they are here.
Hi, Dr. Ambrosone. Thank you so much for this talk. I'm really excited to see what you find in the research combining some of those social factors with these molecular epi. I wanted to hear what your ideas were on some of the translational side outside of modifiable factors. So whether that's towards maybe developing drugs that could help these Black women with these more aggressive molecular subtypes. And if that's the case, if there's any concern about accessing that treatment, if that drug development would actually exacerbate the disparity it's trying to improve. Thank you.
Yeah, that's a very good point. And that's something that we've thought about when they talk about personalized medicine. Is that really going to be available to everybody? The drug development, I'm kind of outside of that. People have talked about, if you think about the role of methylation, can demethylating agents have an impact? But I'm not really familiar with that kind of work. Yeah, you always need to, especially, with grants, you always have to talk about the impact and the so what. As a researcher, it's so interesting to do the research and to see how things work, but it's like, so what? Does it matter? So those are the next steps, and those are the hardest steps for sure.
I think we may have a question online. Sam Smith, if you wanted to unmute and ask your question.
Sure. Let me know if could you hear me?
Yes, we can
Yes.
hear you.
Okay. The question before Sarah's, when I was at ASPO, and I know a couple of the universities or cancer centers in Philadelphia, they had looked at exploring native-born Black women versus Black women who immigrated more recently as a way to address that issue. So it wasn't really a question. I was just throwing that out there, but I understand that sample sizes often can be small.
Yeah. Thank you for that. I'm familiar with a little of that work, but you'll need to dig deep and see what it is about migrating or acculturation into the U.S. Is it racism that could make things worse? That's a good point.
Other questions? Okay, well, let's thank our speaker again.
And thanks to all of you for your participation and attention.
Right, I forgot one more thing. We have a little gift for you.
Oh, is it a plaque? Should I open it?
Yes, please do.
This has been a great visit getting together. We shared an office in graduate school. Joe Freudenheim was my mentor. I worked a bit with Jean Wachowski-West, Wendy. It's just been great getting together with people. Oh, nice. Thank you.
Enjoy.
I lost that jacket in an airport. I miss it so much.
Yeah, so I want to thank everyone for coming. And if the award winners and the Graham family could come up to the front, we're going to take some pictures. And Jean, if you want to come up too.